Meu nome é Marina Oliveira, eu sou gerente de produto da ABD. Eu agradeço a presença de vocês aqui e eu queria apresentar nosso VP na área de científica da ABD, que é Bob Balderas. O Bob trabalha na ABD desde o final dos anos 90, quando a empresa dele, que ele tinha criado, que é a Farmingen, foi adquirida pela ABD, e desde então, há mais de 30 anos, ele trabalha com a ABD no desenvolvimento de novos produtos e de novos dados para a citometria de fluxo. Então, Bob, you are welcome.
Okay, good afternoon, everyone. So in this next 40 minutes, I promise to bring you something that's different and something that is exciting. So let's just think about this for a second. Over the years, there have been many, many technologies that we've developed, the whole program in the field of immunology. And these technologies have allowed us to be able to look deeper into the immune system. And I go back to the one that really had the biggest impact on our field completely, and that was in the early generation of the work that we did in writing the first paper for the CyTOF technology. And it allowed us to move from 18 specificities to 34 different lanthanides, and gave us a chance to be able to look inside the cell in a completely different way. Soon after that, though, flow cytometry began to move forward because there was the generation of many new colors with the Sirigen dye technologies that were used with polymers. And then soon after that, the New York Genome Center developed the technology to bring in and allow us to be able to label an antibody with an oligonucleotide. And since then, there have been many other technologies. But today, we're going to talk about a different technology, a technology that comes back to flow, a technology that allows us to be able to see protein deposition, or the spatial deposition and position of different proteins in a cell or on a cell at the single cell level. Because as we all know, right, genomics has taught us that every cell is different, that every cell is an island, that every cell works and communicates with many other cells. So this is the first time that at the level of 3,000 to 10,000 events per second, we can follow protein deposition inside the cell. But for a moment, for everyone here first, let's go back 50 years. So for 52 years in our field, we have studied and looked at flow from a position of histograms, a position of dot plots or contour maps. And every one of all of our work is always expressed and looked at from that perspective. But if we take the two cells down in the bottom, imagine that every receptor does not have a homogeneous, let's say, expression over the surface of the cell. Some of those receptors are punctated. If we have an activated cell, CD3 or CD4 actually can move to the polar caps upon activation of a cell. So the biology inside has always been there, but when we look at an activated cell, we still see it in the same histogram because we only count photoelectrons. Likewise, though, if we look at the two cells up above, a protein could be, GFP for instance, could be in one or two places in a cell, or it could be in a single place in the cell. But if it all had the same number of photoelectrons, we see those two cells in one histogram. So along came a technology that has changed everything that we do. And imagine, about 25 years ago, when the DIVA technology and the electronics moved from analog to digital, it gave us a chance to do something special, which was called doublet discrimination. And thus, from around the year 2000, we take the doublets and we remove them from our analysis completely. Just imagine there is probably biology in those doublets, and that's what it looks like right there. Or maybe being able to use a flow cytometer to see something different, a change in morphology. And we're going to talk about this image at the end of the presentation. So a flow cytometer that could find that cell in blood, it's an activated T cell that is infected with virus, okay? So let's go on with this story. It was an easy technology that came and was developed about 8 years ago by a scientist by the name of Eric Diebold in the Jabali lab at UCLA. Soon after we met them, we acquired the technology and brought it into BD to be integrated into a spectral instrument. And we thought it was going to be a very easy feat, but in fact, it took us 7 to 8 years to be able to generate the technology you're going to see. At the end, though, it's a very simple process. It's a spectral instrument that has 78 APDs on it, along with 3 PMTs that are used for imaging. And the blue light of that one laser line is split into 2 dimensions, one to flow and one over on the imaging side. And the technology looks something like this, where if you look up at the top, a cell going through this system is interrogated and goes through and is interrogated by 104 beamlets. Each one of those beamlets has a frequency assessed onto it, and that frequency then, as the cell goes through, interrogates a different position on the cell. That information that is translated then into a waveform, and then utilizing an algorithm that's been in our field for about 50 years, it's translated then over into a pixelated 2-dimensional form of a cell. Now, all that is very, very simple, but it looks something like this, where light then is separated by 104 beamlets. Those beamlets then are oscillated with a frequency, as I said. The frequencies then hit the cell in many, many different dimensions, collected as a waveform, and then all brought together with an algorithm to give us what a cell looks like in 2 dimensions, but not 3. So it's an electronic image of a cell. So imagine being able to see all of those different types of cells in the system. If you think about it, maybe even in an easier way, you know, we all have sat on an instrument where we've sorted before. And we look at the one subjective thing that we do in flow on every single sample, and that is we put a gate around a population to assess. And when we're on a sorter, we sometimes sit down to place that gate, and what happens next is we see cells that fall outside of that gate, and you wonder sometimes, should those cells be in the gate or should they not? Well, in this instrument, you can mouse over every single cell going through the flow cytometer and assess what that cell looks like. And then, depending upon what that cell is, we can then move it into the gate or out of a gate. Okay, so just imagine the different kinds of things that you can do with a system like this. And then we collect over on this side all the different cells in what's called an image wall. So as the cells move through, we can watch what happens to any one of those cells, or we can take our mouse and mouse over any place in our scatter diagrams to look at the kind of cells that could be there. Within the context of this system, there are a number of what are called imaging features, and those imaging features are calculated instantaneously at the speed of around 3,000 to 10,000 events per second. Calculations then on each one of these parameters are made, and then decisions, then sort decisions or analytical decisions are made on every single one of those instantaneously. So it's relatively fast in doing that work. And we'll go over some of these as you'll see some of the data that I'll share with you in just a minute. But as I said, doublet discrimination has been something that we've always held on to. It's always brought us great value in being able to make sure that we look at singlets within flow cytometry. But there are many times when doublets could be important, not counting the CAR-T therapies and all the kinds of cell-cell interactions that we actually measure today and induce for therapies, But imagine what could happen in blood. Question would be, in our blood, as individuals, do doublets occur? Do those doublets have biological function? Do T cells bind with B cells? Do T cells bind with monocytes? Are they different by age? Are they different by gender? Are they different by disease? Imagine for 25 years we've thrown them out, but let me show you a story. So within the context of this, if we just look at a simple T cell monocyte interaction of a CD3 and a CD14 positive cell, we look at our dot plots here, and something looks a little different. What's different here is that there is a doublet population. So you have to ask yourself, what is that? Is it a true doublet between a CD14 and a CD3? Is it a cell that has both 14 and 3 on it? But what is in that? So today, with an instrument that has been developed that can actually sort these cells too, we built a panel of around 20 specificities to interrogate both T cells and B cells and monocytes. With the question, are there interactions between T cells and monocytes or T cells and B cells? And if there are there any biases to the types of cells that actually interact? So from that perspective, we break down and look at the biology different, not using doublet discrimination. So with the concepts of different types of imaging features, one of them called radial moment over here, and another one's called eccentricity, we can find the majority of our singlets and the majority of our doublets. So there's a lot of cells in there. So then we have to ask our question, if we stain that whole population and then look up at our CD14, CD3, that doublet population shows up. Now, utilizing different imaging features on this instrument then, we use delta center of mass and radial moment, and you can see that we always get what's called a wing. On that wing, if we look up in this area and gate on those are coincidental doublets. So that means that the instrument reads them as a singlet, but in normal cases, and they would be excluded with doublet discrimination, but in reality, they're just two cells that are very, very close together. If we gate differently then, at this position, you can see that these cells are stuck together. Now, to make sure biologically they're correct, we would use an antibody to a synapse. But if we go down to the T cell B cell interactions as we have here, we still find a doublet here. We move over to the delta center of mass and radial moment on a imaging cytometer. We find our coincidental doublets, and then we move into this, and you find that these cells are stuck together. And if you look very carefully here, there's a little bit of yellow in between those two. That's called coincidence. So when two different cells come together with different colors, we can see a change of color that occurs, and thus we know those cells are stuck together. We can actually sort those cells too, okay? So then utilizing what we're seeing here and what we would normally discard with everything that we do, we take that 20-color panel that we have here and we assess, utilizing just phenotype, all the different types of cell populations that you can imagine using a 20-color panel. And then we just simply ask, in this donor, are there any biases to the T cell B cell interaction or to the T cell monocyte interaction? And the answer is there's biases everywhere. Now, I'm only showing one because there's a lot of other data that's being placed into a publication right now. But usually in most normal individuals, we find with a T cell and a B cell, they're predominantly CD4s and not CD8s. And if we move over to the T cell monocyte, it's a much more, I would say, complex set of interactions, but there's significance in that. And to some of the new diseases, not infectious disease, but in other diseases, we have already begun to ascertain that there are significant similarities between individuals that have the interactions of different types of cells in their blood. So that's one way. But let's take a look at what else an instrument like this can do. One is how we look at the expression of TBAT. And for all of us, we all know the story on TBAT. It's an extremely important transcription factor that helps us understand a lot in T-cell exhaustion. There's also interacting proteins like LAG3, like TIM3, and PD-1 that either positively or negatively up and down regulated depending upon the activation of TBAT. So several years ago, John Weary and Mike Betts at the University of Pennsylvania used a different technology and confocal to actually ascertain the function of TBAT itself. And they looked at that from different perspectives and found that TBAT, when it was highly expressed, was always in the nucleus. But TBAT, when it was low expressed in a non-activated form, sat out in the cytoplasm. Now, one can imagine in a flow cytometer, if we just looked at these cells all together, we would see different populations of TBAT expression, but at the end of the day, if we would be looking at the cell in one dimension of a histogram. So they postulated and built different types of model systems to actually lay down the foundation of TBAT expression for all the memory populations of T cells and showed how that TBAT moved from the cytoplasm into the nucleus, but from one dimension only. Imagine if you could look at all the different other proteins that are regulated along with TBAT itself. So we did this by looking and utilizing flow cytometry. We needed to find a bright color for TBAT to be able to see the two different populations of low expression and high expression. And as you can see, if we look at all the different colors that exist out there for TBAT that are used, they don't all show this. So a heads up to everyone here, not every color on every clone actually tells the same story. So we picked one of the ones that were in the green box. Now when we do that, we actually can look back at stain index. So if we look at and assess all the different, how bright each one of them are, you can see that the bright ones that are in this case always show us the low and the high expression. It's hidden in these and completely gone when the stain index of a given protein is low. So that's very, very important information, I think, for everyone in here when you're building panels, is that not every color works in the same way. So utilizing TBAT on a PE conjugate, we can see in here that the expression of in this sample has both high and low. If we look at two other different ones, you can see where we lose the low and we can get the high, and this one completely loses virtually everything. And if we then bring it down and look in the concept of different colors with just CD3, you can see where the expression of TBAT looks very, very different if we can use a bright color versus what happens when we use a much less bright color, right? Now, so let's just take some of that information and go a step further. So we build a small panel for TBAT to assess other proteins at the very, very same time. And if we look at all of those collectively together across all the different types of memory type cells, or different CD4s and CD8s, you can see the complexity changes depending upon the activation of a given T cell for both CD4 as well as for CD8. If we move back over into the memory populations, I highlighted some of the changes, but you can see those memory populations do change upon activation for a CD4, and when we look over at a CD8 population, they again change too. So now we take all of that complexity and we say, what happens to TBAT within the concept of each one of those memory populations, and what happens to the other additional proteins of LAG3, of PD-1, and TIM3 at the very same time? So utilizing that technology, we can look at the resting cells and the activated PBMCs across all the different memory populations, and you can see where the expression patterns really change. We then move that over and we use a concept on imaging called correlation. So that is when two different proteins move together in concert together, a change of color will occur within the inside of a cell. So imagine now, we have T-bet that sits out in the cytoplasm in a non-activated form, low expression. If it moves into the nucleus, and if we put a dye on that nucleus, we can see then a change of correlation, and we can see that the protein actually moves. Okay? So with that then, we use that kind of technology to look at the difference between the resting CD8s and the activated CD8s as a total population. You can see where the low expression is on this side, and if we look over here, you can see how highly expressed it is on activated cells in each one of those memory populations. Now we go back in just using regular flow. And by virtue of that, we look across for the CD8 populations. You can see the virtual changes that occur for PD1 and for T-bet as they move across. So there's lots of changes inside the cell. But with a traditional flow cytometer, we wouldn't be able to see where the localization of that protein is and ask the question whether or not the protein changes on others based upon where the position of the color is in the cell or the protein. So we looked at PD1 versus T-bet. We looked and brought that over the difference between the resting and the activated C cells for LAG3 and PD1, so you can see the difference there in flow. But then you say, like, if you could know where the spatial location of the protein was to establish real function, it's something that no other flow cytometer in the world can do. So this is how we look at it. If we open the system up and utilize imaging different parameters called correlation, if we look at the resting cells and go all the way across, you see kind of a large peak here. So no correlation is at this end, high correlation is at this end, and if you have activated, you can see where almost everything disappears here and everything goes into a very high, the histogram moves over to the end. But then we just, again, ask ourselves, what does that look like? So if we look at resting cells themselves, we can see that, and if we look at the images that come out from the flow cytometer, wherever there's a red dot, or two of them, depends upon the two dimensions of how the cell goes through the flow cytometer, the red represents the T-bet that's expressed in the cytoplasm. So in this flow cytometer, as we look at each one of those histograms and look inside it, we can see that this is where the resting cells look like with no correlation. As the protein begins to move into the nucleus, it changes color and turns into slightly yellow with some expression of T-bet on the outside. And then if it's highly correlated in this final channel that's out here, it's completely in the nucleus itself, thus allowing us to see where the spatial location of a protein is inside a histogram to be able to establish really how protein expression really works. Now you can ask yourself, again, a different question, that if I look at any one of these given cells, I can look at the expression of other proteins too. So inside that cell, where the T-bet is expressed in the inside of a cell, what do those three other corresponding and regulating proteins also do? And by virtue of that, if we move from the resting cell to the activated, it doesn't change how they look, but the total number of cells changes here. That's high, so just look at that one versus a resting one, where there's very few cells as the cells have moved into the high correlation side, meaning that all of these cells now that sit right here are all in this one now when we move over into that range. So now we take this type of information and we bring it together and ask a different one. In the field of flow, or in every single flow cytometer that are in your laboratory, we can only look at subsets based upon what the histogram looks like. But because now we can look in the histogram and see where the spatial location is, I just charted a few of the different subsets that are here out of 32 different subsets that can be shown. So in flow, we can only see 4. In here, we can see the additional ones that are 8 that are here, up to 32 different subsets. So the power of the technology says we can do many things with it. So let's just do something different with an instrument that has a lot of colors, meaning these spectral instruments. So I did something for everyone just to add a little different dimension, because money is always precious. Our ability to do flow with the errors that we have for each of us when we do flow cytometry is about 5 to 10% between every single sample. So how can you reduce cost of your experiments and reduce the amount of, let's say, changes that occur and the errors that occur inside flow? One way is to use lots of different colors. So let me share with you how to do it. So if we take 3 donor samples over on this side, and we are going to have resting cells, a 24-hour activation with anti-CD3 and 28, and a 48-hour. So we have a kinetics here, and we have 3 samples, okay? So I take those 3 samples that we have here, and using additional colors that we don't use in flow, I barcode them. I barcode them with 3 different concentrations of V500 for CD45 as a barcode, and another one over here with V450. So by virtue of changing the combination of the amount of each one of those, I have 9 different sets of colors, okay? So each population of those 9 have a different combination of V450 or CD45. Now I take those and I add, after I stain or dye my cells, it takes about 5 to 10 minutes to do that work. Then I use a panel over here to address the biology. Now using that, I then go through with the imaging parameters. I find my singlets that are in here, many of our doublets that might be in this range. And remember, I barcoded these with different dimensions. So these have the resting cells with CD45, then in here, different amounts of CD45. So the intensity goes up. I then barcoded them with a different one for a V450, which is an amine binding dye. And now I can see 9 populations, all 9 together in one tube, okay, that have been stained by that cocktail that you saw. By utilizing a technology like that, I can then go back into here, and on a flow cytometer, I can put a gate around all 9 of those populations and then look inside each one for the basal levels of TBET, for the 24-hour stem, and for the 48-hour stem. And then I can take that information and take each one of those as I put a gate around it. Remember, this was stained in one tube. So it's about 1/8 of the total amount of color and reagent that I needed to do in one sample. I did 9 of them in the same tube. So you can see that I can put a gate around this population and look at all the biology across that panel. I can put a gate around the 24-hour stem for donor number 1 and see that. And I can put a gate around the 3rd population over here that was barcoded and to see what the biology looks there. And you can see the changes that occur over time, all together in a single sample. Now imagine all of that flow, and that we look inside it and say, where is the expression in what part of the cell? And then I simply then put different gates around the correlation in here for TBET, as we see low, medium, and high. Low, and then in the TBET high, where almost all of them are over here. And you can see that in the low correlation, the TBET is on the outside with small amount going inside the cell. And if I move over here, the TBET's completely gone and it's 100% inside that cell. So it shows you how to use an instrument that gives you a chance to think about the protein localization, the spatial location of a given, at least one, if not three different proteins at the very same time inside a cell. So I promised you in closing here that I would look at it one more time. And let's go to the last one, which is right here. I thought it might be apropos to bring something on infectious disease in here. So I have, we work with a team in Mexico City at an institution called CNA. And it's connected to a hospital as well as a research institute. And it's predominantly, virtually 100%, dedicated to individuals that have either HIV or HIV and TB at the same time. And it's the second largest one in Mexico City. And to show you the breadth of problems that they have, they run about 1,000 CD4 counts a day. They do it 7 days a week and 365 days a year. So it's a big problem. And it's the second largest HIV program in Mexico City. So I took this technology down and I asked Gonzalo Salado if he was interested in trying to use this technology in his field. Never used it before. And he brought a whole series of scientists into the room, and we just had a discussion about what the technology could do. So we all know this, right? How viral replication occurs. We know it very, very well. Whether we can stop it, that's the other story, and cure it. But what we do know is that when an activated CD4 cell, when it's activated, there's a morphological change in those cells. And when that morphological change occurs, we see a distal uropod that's up at one end. This is also where homing occurs. So if we would have this is an EM picture, but if we would stain the cell with all the chemokine receptors, the majority of the chemokine receptor expression sits right at that part of the cell. So it kind of pinches off and looks like a bell shape, even though this is a EM picture. So we did some simple experiments inside where we took normals, a whole series of them, and then we took individuals that had just come into the clinic that had not been on drug. And we simply took their blood over a series of them, and to bring consensus and to make sure the experiment was done right, we activated both populations with a trigger with PHA and IL-2. So we wanted to see the activation in normal cells too. So after that activation then, and utilizing what we can see based on morphology, we can see that there is activation in the normal cells, but the activation looks completely different in all of the HIV patients that we looked at. And then we simply ask what was in that population. So imagine looking at a disease from a different point of view that we normally don't use. A point of view where we can look for cell surface receptors, we can look for protein expression in a cell, but can we find cells that are actually infected too? So by virtue of the technology that I shared, bringing imaging in at the same time at the speed of around 10,000 events a second, that's what we found. That is an activated T cell found in the blood of an individual. The yellow is CD4 with the brightest possible color we can use in flow because there is downregulation. I then sorted those cells and then fixed them and permed them and used an antibody to P24. And that's what the green represents there. So in the context of different individuals, we could see many different kinds of shapes and sizes. You see that this one looks like the one in the previous picture. This one a little bit does, because this instrument only looks in 2 dimensions. So although I sorted the cells out on a dimension of light loss, which gave me the morphological change, if the cells go through the laser this way, the very first time, and I collect them all, If some of them go through this way the second time, then they don't capture the same imaging look. So the cells look a little different in a post-sort, let's say, purity check. But as you can see here, we can actually even use that technology to actually look at the amount of infectivity that could possibly be in certain kind of cells too. So it's a technology that's going to change everything in our field. And in about a month and a half from now, Mark Davis is going to show you a rebuild of our understanding of Treg biology. He showed it recently in Cold Spring Harbor, and it's in review right now at Nature. But I think once it goes out there, it's going to change the way we all view immunology. And it's going to change the way that we think how we use a flow cytometer in our field today, that if you can bring an imaging compartment into what we can do with many colors of flow, then it's going to allow us to do one thing that the genomic world has told us for a long, long time, that every cell is different, that every cell is an island, that cells communicate together. The only difference is we've never been able to see that at a protein level. So for the many scientists that I work with every single day, it's allowed us to take a technology not only into the field of immunology, but just imagine in the field of cell biology that have looked in the use of a microscope for many years, the one thing they've never been able to do is isolate the cells they need, like we do in our field. So for all of that, different things, there's many people that would like to share a lot of the work at Emory University with Rafiq and with Susan, who was here in Esper's laboratory for many, many years. But a lot of individuals that have contributed in the development of a technology that truly is going to change the world in our entire field. So for that, I say thank you, and to also thank my team from BD Brazil too. Thank you very much. And if we have a chance for a question, that'll be fine.
Se alguém tiver alguma pergunta, se quiser fazer em português também, eu posso ajudar a traduzir. Fique à vontade.
Okay. If not, I'll be here for a while, and you can come and ask the question. I'll try to answer it for you. Thank you.
A gente vai estar aí amanhã também, vai ter um representante da BD aqui. Esses equipamentos que o Bob mostrou com essa tecnologia, a gente já tem 5 no Brasil, o 6º está vindo. Eles estão disponíveis em São Paulo para demonstração. Então, se vocês quiserem, tiverem interesse de passar a amostra de vocês nesses equipamentos, a gente pode agendar com esses clientes a demonstração nesse equipamento para vocês conseguirem fazer uma citometria em que vocês seriam capazes de fazer o gating ou o sorting das células com base nas imagens que vocês visualizam no citômetro. Então, ele gera uma imagem para cada uma das células que passa pelo fluxo do citômetro. Tá? A gente também está com promoção de reagentes até setembro. Então, se vocês quiserem receber mais detalhes dessa promoção, a gente tem ali um QR Code na mesa onde a gente está expondo. Passem lá para escanear, se cadastrem, e aí a gente manda um e-mail para vocês. Obrigada, parabéns pelo evento e boa continuidade de evento para vocês.
Good afternoon, everyone. We'll start the second session, and we'll be discussing now vaccines. We have a great team of speakers, and we will start with Dr. Elder Nakaya. Professor Nakaya holds a bachelor degree in biological sciences and a PhD in biochemistry from the University of São Paulo. Has done postdoctoral periods at the University of São Paulo and Emory University, and currently is a senior researcher at the Hospital Israelita Albert Einstein, and a professor at University of São Paulo. Welcome, Professor.
Hey everyone, So I'd like to thank so much the organizers for inviting me here to talk about systems vaccinology, because this is how I met Ricardo. And this is how AI would animate the scene of Edward Jenner vaccinating the Gardner boy. Well, if Edward Jenner was alive today, he would probably be doing vaccinology this way, because now, like, everything is data, as you saw in the previous talk. So it's a lot of data. And if Edward Jenner was alive today, probably Ricardo Gazzinelli was going to be his boss, because, and I'll tell you this because Ricardo is connected to everyone, and that's a fact, because I download all his papers over the last 30 years. So I check the co-authors on all these papers. So probably everyone here is there among these 1,414 authors. So he's highly connected. And that also has to do with the talk that I wanted to give about systems vaccinology. So why systems vaccinology, and why this is relevant for vaccines? Well, if you take a group of people and then give the same vaccine, do you agree that not everyone is going to respond the same way? Some are going to be protected, some are going to have adverse events, and some will die or is not going to be protected and is going to eventually die because of the infection. And that happens because every single one of us has a unique immune system. We have different molecular factors, malnutrition, stress, everything influences the outcome. And nowadays we are able to measure all these biological components together, so we can create a network like this with a lot of data. And then we have to in order to make sense of this data, we have to use network science and precision medicine to try to find out what is the smallest part of this network that can predict an outcome. And that's a misconception when we're dealing with omics, because we are trying to focus on the biological components that change the most. So, for example, if you have a list of genes or proteins that are altered after vaccination, we will focus on the single gene or single protein that is changed tenfold. But biologically speaking, this single gene in the periphery of a network may be less relevant than a group of genes that are highly connected but are with a subtle change. And that's the idea of network science, because you have to take into account not only the levels of the proteins or the genes, but also how they are connected in the network. And that's influenced the response. And especially for vaccination, this is not only looking at the host site, because you also have different types of vaccines, you have different routes of vaccination, adjuvants, everything influences the outcome. So you have to take into account not only the genes inside the humans, but also how you're giving the vaccines to the people. And that's the precision vaccinology that I'm talking about. I started in this field when I went to do a postdoc in Bali Pulender's lab. And in that time, what he was trying to do, there was no RNA-seq in the time, and was to create a chip, something that we can measure signatures that is going to be associated with vaccine-induced response. And that's what I tried to do when I was there. And then we come up with this term called systems vaccinology that tries to explain how we can use computational modeling, integrative biology for studying vaccines. And then later, when I was already here in Brazil, I tried to go one level ahead to go to network vaccinology, and I'll explain briefly what it is. But when I was a postdoc there, I tried to investigate why influenza vaccine, the same vaccine, gives different immune response according to the age group. So if you give the same vaccine to children, adults, or elderly people, you're going to have different response. And then I tried to investigate the mechanism behind this. But I'm going to focus only in one paper that shows some genes that were associated with antibody response. So these genes were altered during the first week of vaccination, and they can predict antibody response 1 month later. And the intriguing part is that one of the genes that was there was toll-like receptor 5, which was associated with antibody response. And as you know, TLR5 is a receptor that binds to flagellin, which is a bacterial component. It has nothing to do with inactivated influenza vaccine, virus vaccine. In that time, keep in mind, that was 2009, 2010. Nobody talked about microbiota. And the first thing we did was to see if knockout mice for toll-like receptor 5 would have a different immune response. And yes, the response was lower than wild type. And when you try to find out what is the ligand related to this, how can you test this? By treating animals with antibiotics or buying germ-free mouse. And then when Jason did this, you can see that the amount of bacteria in the stool decreases in these mice. And when you vaccinate them with influenza vaccine, now it shows that the immune response was diminished. That was the first time that someone established a link between the gut microbiota with a vaccine-induced response against a virus. And that was very fun because later they showed that the humans treated with antibiotics also have different responses to vaccinations because of gut microbiota. And that doesn't work for all vaccines. Strong vaccines such as live attenuated yellow fever vaccine, gut microbiota has no role on this. But inactivated flu vaccine, we could see this. And then I want to show this because I was still working with Bali when I showed this. I showed to him that there was a signature of platelets in the influenza vaccine that was correlating with long-lasting antibody response. And remember, vaccines has to induce not only high immune response, antibody levels, for example, but long-lasting response. And one of this group of genes that were associated with this long-lasting response was platelets. And that was weird because why this would be there, right? And then I literally forgot about these results, but then I found out that Bali was still working on this, and he had a perfect model because when he vaccinates mice, no, this is human, with H5N1 vaccine, there was no response. When you use the adjuvants, you induce a very strong and long-lasting antibody response. So it was a perfect model to study this platelets role. And what we saw is that that signature that I showed was also confirming this model that's showing that platelets seems to be important for inducing this long-lasting response, and the inactivated vaccine without it was not. And then what Bali did was a lot of experiments using, for example, TPO, thrombopoietin, that activate megakaryocytes that are the ones that produce the platelets. And he shows that once you put TPO, the antibody response is higher and remains high for a long period. And then he showed that there was no physical, the physical contact between B cells, the antibody secreting cells, and the megakaryocyte was important for giving this long-lasting response. And then he showed that bone marrow in humans, you can also stimulate with this TPO and megakaryocytes. And that was published this year in January, and I was glad that I helped with this paper too. But that's just to show how long it takes from systems biology to become a paper like this. 10 years. So 10 years of validations, a lot of experiments showing that the mechanistic insights could be checked elsewhere. And since then, my lab has been working with the same approach, systems biology, but now applying to different disease, especially infectious disease. Here is one example, but I'm not going to talk about this paper that shows the importance of long non-coding in this vaccine-induced immunity. This one was a collaboration with Daniela Ferreira from now in Oxford that tries to investigate why people that have pneumonia have more susceptibility, no, people that have influenza has more susceptibility of having later pneumonia. And that is already established in epidemiological studies. In children is the number one cause of death. And one important thing to know is that in order to have pneumonia, you have to first have your nasal mucosa being colonized. So what we are interested was to try to model or try to understand how influenza virus make you more susceptible of having pneumonia. And that is very hard to test this unless you have a challenge model. And that's exactly what she has. So in that time in Liverpool, she has a human challenge model where they can infect humans with pneumococcus. And here in Brazil, we helped analyze with this data. So this is to show how you inoculate the bacteria, and then you get the nasal wash, and then you do the transcriptomics, and also you check if the person was colonized or not. So who would accept to be part of this study? What if I give you 1,600 pounds for this? Now you see like some people would accept. And nobody got sick or died from this. And this is to show that now we have a perfect model, because then you can infect someone with influenza. Of course, not the wild type influenza, but the live attenuated influenza vaccine given in the nasal. And after that, you can challenge with the bacteria. And then you can measure the transcriptomic change that happens over this. So we saw the genes that were affected during this process. We compared the groups between the ones that were previously infected with influenza or not. But then I'm going to summarize in this model that's try to explain what we think is happening. So if you have no previous virus infections, and then you get colonized, neutrophils go there, macrophages actually are here. Monocytes, neutrophils call the monocytes to go there and then start the clearance. But if you have first an influenza infection, there is a local inflammation there, IP10, that impair this migration, the dysfunction of neutrophils that is going to reduce monocytes and migrating to the nose. Of course, there was a lot of flow cytometry and transcriptomics to prove this. Patricia in my lab, we were part of this Ebola vaccine consortium, and what she was interested in was on the side effects of Ebola vaccine. So Ebola vaccine is a wonderful vaccine in terms of immunogenicity. It gives you like 100% protection, long-lasting antibody response, but the problem is that the vaccine is very reactogenic. So it can give you even arthritis for especially Americans and Europeans, but none from Africa. And what we want to understand is why some of the vaccinees would develop arthritis after vaccination. And we use random forest, a machine learning approach, to try to identify the genes that could predict who's going to have arthritis a few weeks later. And I'm going to explain how machine learning, how random forest works, to show you that it's not that difficult to understand. So imagine that you have dozens of patients, and in this case, only showing 9 people that don't have arthritis or 9 that have arthritis. And then imagine now a table with 25,000 rows, which are the genes expression. And what random forest does is picking one gene, and then you can place the people that had no arthritis or had arthritis, and then come up with a cutoff that separates most of the blue dots from the red dots. And then you put this information here, and then you repeat this with another gene, and you put this information there, and then you keep doing this until you separate everyone that had arthritis or not. So that's a decision tree. And the cool thing is that random forest, you don't use the whole data set to create one decision tree. What you do, you get a random number of subjects and a random number of genes, and then you create these random data sets, and then you create a decision tree. And when you repeat 1,000 times this process, and you have a place with 1,000 trees, what is the name of this? Forest. So random forest is very easy to understand because then you can count the number of times that the gene appeared in these decision trees, because that's going to give you the importance of the gene in predicting an outcome. So when we did this, we found out what were the genes that could predict who had 2 or more adverse events, and we can also identify for each adverse event what are the genes that were predicting that adverse event. And then we can create a network showing the genes and the adverse events to try to understand or predict who's going to have severe adverse events for the Ebola vaccine. Recently, this was December last year, we published this paper with transcriptomics of acute infection of yellow fever. In 2018, in São Paulo, there were an outbreak of yellow fever, and then we collect the blood of these people, measure the transcriptomics, and then analyze the change that happens and try to understand why some of the people will die with yellow fever and some will not. But the cool thing that we left out of this paper that we are going to publish now is to compare the acute infection with the infection caused by the live attenuated yellow fever vaccine. Because the idea was that people claim that the live attenuated influenza vaccine, yellow fever vaccine, is a mild infection. It changed basically the same genes of the acute infection. And what we are seeing is that that is not true. Of course, type 1 interferon, it changed the same, not in the same magnitude, but there were a lot of genes that were unique to the acute infection that had nothing to do with the vaccination. And we are also doing this Vaccinology knowledge graph because we can also download 45 million papers and then ask the machine, the computers, to read the text. We can see, for example, relationships that were described in the manuscript. So vaccine A induced the gene X, and then we can create graphs from this. So for example, we can take all these 45 million papers, ask a machine to read this, create this knowledge graph connecting genes, drugs, and disease, and then put in humans to try to get insights and hypotheses from these knowledge graphs. So this is the knowledge graph of all the commercial vaccines that we can use in humans and the genes that are related to these vaccines in the literature. This is not experimental from transcriptomic data. This is from papers. And then we can zoom in and try to start seeing the genes that were affecting one or more of, were related to one or more of this vaccine. We could group these based on the vaccine routes or types of vaccine used or not of adjuvants. And that's part of the computational tools that we have been developing over the years. In my lab, we are actually on page 2. But I want to finish with this unpublished paper tool called Immunovis, because we are going to use this for vaccine knowledge as well. The idea is that neural networks works really well for classifying image. So if you put the image of a mole, it can tell you if this is melanoma or not. So it's done deal. This is, works really well. But the problem is that if you take now the sample and extract the RNA and have now a table where you have 25,000 rows and then hundreds of columns, and then you use the same neural network, that's not going to give you reliable markers that can predict populations that are very diverse. And that's not because the machine learning method doesn't work. It's probably because biology is more complex than this. You have different sets of genes that can drive or can get you cancer. But and that happens in a global scale when you do like an experiment with one cohort here, but you cannot validate in another cohort there. So the idea is how can we reduce the complexity? And one way we thought was to use computer vision. The idea is to put these genes together in an image. So we transform this into an image, and then we use neural networks to classify the samples, because neural networks works really well for image. So we have, for example, the expression data and pathways, and then we can place the genes based on the similarity of expression patterns in a tissue or the similarity in biological process. So each dot, which is going to be a pixel, is going to be a gene group according to the tissue. And the idea is to have like a colored book, empty, where you can paint with the expression levels of each individual. So imagine that we apply this in cancer. Imagine that this is 3 different samples of normal tissue and 3 different cancer samples. Doesn't matter what is the genes related to normal cancer, normal samples. What matters is that in the center is going to be bright. Here in cancer, it doesn't matter which marker you're actually activating. What matters is that in the right corner is going to be bright. So that's how you reduce reduce complexity. And then we apply this, for example, as a test in the skin. So this is the color book empty for skin, and this is the skin of 3 normal people, and these are the skin of patients with cutaneous leishmaniasis. You don't have to be a computer to see that there is a huge difference here. And then we can do the same thing with bloods. These are the pictures of 6 different people that have health controls, and these are people with acute infection with chikungunya, which is very similar to dengue, very similar to malaria. And here's another example where we took 4 independent tuberculosis studies and that have samples that were active tuberculosis or latent tuberculosis. And then for each sample, we create an image, and then we train the model based on 3 independent studies, and then we test on this 4th independent study. And then, as you can see, the samples that were actually active TB were predicted to be active TB. And the ones that were latent TB, they were predicted to be latent TB. So we are now going to apply this to citizens vaccinology studies to see if we can use this approach to classify who's going to have high antibody response or high T-cell response after vaccination. So to finish, I would like to say that we are under AI revolution, and that is going to affect everything, all scientific fields, and of course vaccinology. And for vaccination, you have plenty of usage for AI, which is actually already ongoing of most of this. And the problem is, so when we're going to have vaccines against everything, right? So it's very hard to predict when. Imagine that if we could go back in time and ask Pasteur when we're going to have mRNA vaccines, he will never be able to guess when we're going to have. But the same thing, I cannot tell you when we're going to have vaccines against all the diseases, but the only thing I can assure is that it's going to be during your lifetime, and you're going to see this happening. And with that, I'd like to acknowledge the sponsors of these studies and the team of my lab that is not working. Yeah. Well, that's not working, the last slide, but thank you, guys.
We have time for one question, if anyone has one, would like to.
Hi, Elder. Great lecture.
Hi, Gregor. Thank you.
I was curious in your last tool. You just use the data from the RNA-seq and you do the dimensionality reduction, and then use the AI to check for patterns.
Yes. Yes. We basically we are now trying to figure out how to better capture this as an image, because, for example, P53 cannot be 1 pixel for cancer because it has a weight that we have to take into account. So The first round that we did was just treating every gene as a pixel and then putting there in a dimension reduction way with UMAP, Disney. And from that, we use computer vision, treating this as an image, not as a vector of numbers.
And do you think you can transform like not this kind of data, but data like if people have pain or if people have headaches, and to transform this in numbers, and then you apply to this model?
I see. It's to be also parts of the pixels, right? Yeah, could be. That's a good idea.
Great.
Yeah.
Perfect. Thank you, Dr. Nakaya. We will continue. We will have now Dr. Ricardo Gazzinelli. Professor Gazzinelli is a veterinarian and a PhD in biochemistry and immunology by the Universidade Federal de Minas Gerais. He has several national and international accomplishments and is a member of our Brazilian Academy of Sciences. Currently, he's the coordinator of CTVacinas, the Center for Technology in Vaccines, and a full professor here at the university and a senior researcher at Fiocruz-Minas. It's a pleasure to have you.
Okay, good afternoon. Well, I will speak about this. Most of my talk is about the COVID-19 vaccine that we have been developing in here at the CTVacinas, UFMG in Fiocruz-Minas. And I'll give a brief introduction saying what's the CTVacinas. The idea of the CTVacinas is take projects that have proof of concept at UFMG, Fiocruz, or other institutions from Brazil and try to develop the prototype and something that could be used in human. And then we are not a factory, but we'll try to transfer to the industry. And we have few cases that we have done that already. So this is the model. And all started, we have this, we're part of the national, I coordinate the National Institute for Science and Technology on Vaccines since 2005. And we have, in 2016, we started our, we were a virtual network. And in 2016, we have we were giving from the university our physical lab to do this translational research. And then during the pandemic, it was really the time that we really grow. And and the main problem and become very obvious that Brazil was highly dependent in imported material for diagnostic, for vaccines, and everything arrived late in Brazil. And that was a real problem. So the Ministry of Science and Technology decided that Brazil needed to have a national center for innovation in vaccines. So I like to say that we have an almost complete ecosystem for vaccines. We have the universities that have various projects in vaccines. We have lots of the clinical trial of vaccines that came from outside were done in Brazil. So, and we have the big factories of vaccines. And we have a big, the SUS, the health service from the government, that distribute the vaccines very fast. So once the vaccine arrived, we have a big part of the I think it was 70–80% of the population vaccinated in a very short period of time. But we don't have this some places that make this transition to the university and the phase 1 clinical trial. So no infrastructure, no expertise to produce clinical batches for phase 1 clinical trial, limited interaction with the regulatory agents, and a limited experience of phase 1 clinical trial in general. For vaccine, I think we have 1 or 2 cases in Brazilian history, and they were done in partnership with outside. And then we have a little exchange between the pharmaceutical companies, and that's what we try to cover during once this institute was created, with funding primarily from the Ministry of Science and Technology and from the state of Minas Gerais and from UFMG. So, and then the big problem, we all know, I don't need to give an introduction about COVID-19, you all know about it. And the big problem we have on the vaccine for COVID-19 is the big number of mutations and the specificity of the neutralizing antibodies. So here is the spike from Delta, and here in red are the mutations. And then when you go to the Omicron, you see that there are so many mutations that basically the neutralizing antibody elicited by the ancestral variant doesn't really work at all. So the idea is, which at the beginning was somewhat polemic, was to make a vaccine that was basically direct to improve T-cell mediated immunity rather than neutralizing antibody. And it seems to be crazy, but then with the emergence of all variants, it seems that was not that crazy, because it's very much difficult to escape to the T-cell based immunity, because you would need to have mutations in different peptides that are the antigens simultaneously, and at the point of view statistically that doesn't happen. So that was the idea. So going back, what are the problems we have today is the continuous emergencies of the variants, the delayed development of the updated vaccines, and the immunological imprint, because most people were highly immunized for various times with the spike of the ancestral variant. So just to give one idea, here's the emergency of all variants in Brazil till August 2024. And here is when the vaccine came. So this was the Wuhan vaccine, which worked somewhat for the first variant. It wasn't that efficient, but worked reasonably well for the Gamma and Delta. But you see, the Beta variant was already gone. And then after that, when the updated vaccine arrived in Brazil, you can see BA1. Here is when BA1 disappeared, BA4, BA5. Here is when BA4 and BA5 disappear, and there's XBB.1.5, and here when that disappears. So we never got an updated vaccine for the right variant. They were always late. And this is just to explain for non-immunologists that what is the immune imprinting. So most people have been vaccinated, have received 2-3 shots of this. And then when you come with the spike from the variants, you increase the response to this part, which is not the part involved on virus binding and where the neutralizing antibodies bind. So this is, even when you immunize with these different variants, you have a high response to the original spike, but you don't have a high response of neutralizing antibodies to the RBD, which is the region that binds to the host cell receptor that's necessary for the virus to invade the cells. So the idea is I think if you do a T-cell based vaccine, the idea is that you're going to have you're going to work like the immune system, and that's the way the immune system works. You have an infection with a pathogen, and the first response that comes up is a T-cell response. And after that, it controls the virus, is clearly not as efficient as the neutralizing antibody, but it gives time for the neutralizing antibody to develop, and that's when you clear the infection. So if you if you have a vaccine that favors T-cell response, the idea is that start working earlier, and then you're going to have early production of antibody to that specific virus, and that's how we think that these T-cell based vaccines would work. So to do that, we made a chimera protein that has the nucleocapsid. It's a very immunogenic protein of the virus, and it's very conserved against the different variants. So we use that as a main part of the chimera, and we use a part of the spike protein that we think is important, because if you have primed T cells for the spike, then it will be a helper for the antibody production will be faster than if you don't have. So the idea is to have a primed T cells for both the nucleocapsid that was conserved and a segment of the spike protein. So this was our first paper that came with the proof of concept. Here is just to show that the response and the protection was dependent on T cells, so we immunize the mice, and then we deplete CD8, or we deplete CD4, or we deplete both CD4 and CD8, so the mice were no longer protected. Here are the mice that have been vaccinated and are fully protected. And here is to show that if we do serum transfer, again, we don't protect the mice against infection. If you get a vaccine, at the time we use AstraZeneca, then if you transfer the serum, you have full protection. So this was clearly a T-cell based protection against the virus. And here, so the question when we went to submit our paper, our documentation to Anvisa, they really asked if the vaccine would protect against the variants, and that was our idea. And here is the results from using the hamster, where we use the Wuhan as control. We have a nice protection. Here is the lung structure versus this on the non-vaccinated versus vaccinated. We use the delta, and we see similar protection. This is viral load. And here, again, is the Wuhan on the transgenic mouse that's highly susceptible to infection, but also protect against the Omicron, which is this a variant that the spike is quite different. Here is the lung that's totally conserved the lung structure versus the mice that were not vaccinated. So the vaccine works against the variants. So then we have to do this transition. We received funding for that, and that was to develop the process to produce the vaccine and then to transfer to a company. So we developed the whole process to produce this chimera in a good laboratory practice. We were audited by this institute at Brazil, that's the Metro, which have this control, the process of factories and some of the laboratories. And then we send this to, we also developed the adjuvant. Initially, we were using the poly-ICLC Hiltonol, but they have some problem of analytical data to study the stability, so we change this to the CTVac, which is a copy of the Adavax, that's a squalene called MF59, I think. And yeah, I think it's ME4. Anyway, basically the patent was expired, so we did all the comparative result, comparability studies. We did stability and showed that the activity was similar. So we used that as an adjuvant. And sorry, this is in Portuguese, but we did all the controls, contaminant, showed that the protein is not degraded, and so on. And did also all the tests for stability, primary, secondary, tertiary structure, degradation, look for the size of particle, all that was done, which was required by Anvisa. So with that, we got the approval by the ethical committee, National Ethical Committee and Anvisa, and we started a phase 1/2 clinical trial, double-blind, randomized, control with the active comparator. At the time, we used the AstraZeneca vaccine, and we looked for safety and immunogenicity. This was a lot done by Greg, but really he coordinated a big group. And Elton Santiago is the director of our clinical center. And we use, on the phase 1, we use groups of 9 patients with 3 different doses, and then the control comparative, the comparator was AstraZeneca, again, if you add here, 9. And again, we looked that there was no side effect that was worse than the AstraZeneca. In general, was actually better, although we couldn't do a statistic because the number is small. So based on that and immunogenicity results of this phase 1, we selected the 60 microgram dose for protein, and we started the clinical trial phase 2 for patients, for individuals from 18 to 85. And each group, we have 160 individuals, and we collect samples 0, 14, 28, and so on till 1 year. So the follow-up of the patients were 1 year. And we have completed that last month. So again, I won't go through this data, but basically the phase 2 clinical trial, we confirmed all that there was no worst side effects. Then, and the phase 2 clinical trial, we used the RNA vaccine from Pfizer, which has 2 variants, Omicron, 2 subtypes of Omicron. So I think it was BA4 and BA5 at the time. There was the vaccine that was used at the time. So basically, there was some small difference, always in favor of SpinTech, but never statistically different. So here is the intensity, and here is the cause, whether it was not related, not probably not, possibly, probably yes, and certainly yes. And this is the results with the community, and then with the SpinTech. So going to the antibody response, we see that we have, if you look for the Pfizer vaccine, induces a higher level of antibody to the S Wuhan and S Omicron, as we expect, because our vaccine doesn't induce a lot of neutralizing antibody. And here we use look for the antibody against the nucleotide. We have a much higher response from induced by our vaccine as compared. We didn't expect to have a high response induced by the Pfizer. And if we look for the chimera, again, our vaccine induced a much higher response to the chimera. If you look to the neutralizing antibody, again, here's the Pfizer levels of neutralizing antibody. And here, this is the Wuhan, and this is the Omicron. And as I said about the immune imprinting, you can see that despite the fact that people were vaccinated with an mRNA vaccine that has they codify the spike from Omicron, you can see that the response to the Omicron spike was much lower than the response to the Wuhan. It was at least 20 times lower. So again, favoring this idea that the response to the ancestral spike that people were vaccinated multiple times in the beginning of the pandemic really switch and make the response stronger to the Wuhan. And then our vaccine, you see there was a small increase. It was statistically, but not compared to the Pfizer. And if you go to the Wuhan, there was not really a difference. However, when we look for the T-cell response, you can see that the response to spike was similar when we compared our vaccine with the Comirnaty. There was here the levels of interferon gamma. And when we look for the nucleoprotein, then the response was higher for induced by the spin tech. And then if you look for the Delta is not the strain, but it's the difference, then was much higher for our, induced by our vaccine. So in general, as we planned, the T-cell response was higher when we use our vaccine versus the Comirnaty. And this was the result because when we start doing the clinical trial and submit to Anvisa, they basically said the only biomarker of protection that you can use is the neutralizing antibody, despite the fact that the whole rationale of our vaccine was a T-cell based. And we're like, oh gosh, let's forget about it. But then we got this result that makes us very excited. We look for people that got infected, that were vaccinated and got infected. And if you look, 24 people that were vaccinated with mRNA vaccine got infected, and 15 people from that received the spin tech got infected. This is the curve of cumulative infection during 1 year, and this is the mRNA vaccine, and that's the spin tech, the chimera. So we have a lower, if we do the statistic, it wasn't a clinical trial to prove that, but you show a tendency, and that was sufficient for us to argue that we should go for a phase 3 clinical trial based and do, instead of immunobridging, do a efficacy study. And that's what we're discussing now with Anvisa. And that's the other thing that was interesting. This was the variants that the people got infected from the Comirnaty. These were the variants. Here is small, but it's like JN1, KP, and so on. Each color is a different subvariant. And this is the one from spin tech. The problem is, when we look at spin tech, a lot of the strains were unassigned. Here, only 1 or 2 patients were unassigned. And we say, well, why they're unassigned? And we said, well, maybe the obvious reason was because the viral load and the circulating RNA was very low, and that's the fact. So here is the result of the RT-PCR, and you can see the unassigned from the level of virus is much lower. This is the number of cycles. So depending, if it's low, you need to do much more cycle to have a positive result. So that tells us that probably this vaccine was, in addition to have less patients infected, maybe has a lower viral load. I need to make make it very clear here that these studies were not, we didn't have enough number of patients or volunteers to prove that this is real. So that's why we're now planning to have a phase 3 clinical trial with, I think, with 5,000 patients, around 5,000. But, you know, it shows a clear tendency. So this was the roadmap of everything we've done for developing this vaccine, and we complete the phase 2, and we're discussing with Anvisa to start the phase 3. But here is some advantage of our vaccine. There was, well, our rationale is that won't need to be updated. It's low-cost production. We have all the infrastructure available and expertise available in Brazilian pharmaceutical companies. It has a high stability, at least 24 months at 4 degrees, and maybe 1 or 2 weeks at room temperature. So putting all together, this would be a nice product that will let us go to distant areas that have not so good infrastructure. So that's, and in addition that it will make the Brazilian economy to roll. So finally, I don't know how is my time. I still have 5 minutes, and I'll be very brief. So currently, we are developing a vaccine for malaria, and this is Ana Clara, Ana Luisa, and Irene from São Paulo that are working. Irene was developed the vaccine on the experimental model, and Ana Luisa and Ana Clara, myself, Santuza, and many others from the CTVacinas are really working on all this process to make all the documentation. This is already submitted to Anvisa. Believe it or not, it's 9,000 pages. I don't know how much got to that amount of information, but we are then waiting for approval to start the phase 1 clinical trial, and we hope that will happen this year. We are also developing a therapeutic vaccine for leishmaniasis, and Bianca and Marjorie are leading the project, but again, lots of people involved, Ana Paula Fernandes. And this vaccine is based on one antigen oh gosh. One antigen that was used in this dog vaccine, that was a product that we have been selling in Brazil since 2008. It was a really big success, probably on the pet industry. It makes each year the whole chain of from the factory to the move like about 200 million reais per year, and we have immunized millions and millions of dogs with that vaccine. So we use the same antigen, that's a mastigote-specific antigen. And here we found another antigen. We wanted to make it more structure more complex, so we will be recognized by more numbers of HLA. And so this is a chimera, and this is also in the stage we have done safety tests in mice, and maybe we'll be able to submit it. We have done all the process already in good laboratory practice, and probably move this to Anvisa on the next months. And we use here as an adjuvant the Hiltonol, which is a poly-ICLC, which is a double-stranded RNA. The same for T. cruzi. We have a chimera that we use an antigen from triple mastigote and one from a mastigote. This vaccine, again, it will be a therapeutic vaccine. We use the poly-ICLC as an adjuvant. And this is still on a more initial stage of development. Then we have a platform for RNA vaccine. I won't talk about it, but I think Santuza must be here somewhere, I hope. Oh, here, she's here, and she'll show some of the results. We have a nice vaccine that worked for dengue in the experimental model and for the current H5N1 influenza strain. So but we still have to learn how to scale up this mRNA vaccine. So we need collaboration for that. But we have developed the whole platform. We use a lipid composition that's different than the Moderna and the Pfizer. So here is the portfolio, the COVID, malaria, visceral leishmaniasis, the stage of development. In dark green is the stage that we have accomplished. And, that's it. So I'd like to finally thank our board, which is Ana Paula, Elton, Santuza, Renan, and myself. And we have other researchers that are here, Gabriel, and so others, Giselle, and so on. And this is our group, which started in 2016. We were about 10, 20, but during the pandemic and working on this project, we now have about 80 to 90 people working, and we have different platforms and developing vaccines with Sérgio Costa, Irene from USP, and some products that have been initially developed at UFMG. And that's it.
We will have time for one question. We will have, yeah, come, could you?
Hi, Ricardo. Fantastic work from the whole center, from the SpinTech vaccine. So I have a question about that. So you think that the vaccine is protecting primarily by CD8 T cell, cytotoxic T cells? Do you think so?
Yeah, we did some experiments with the B cell knockout mice, and it's a B cell transgenic mouse. Mm-hmm. And when you use that, they are partially protected versus the wild type that's completely protected. So maybe the idea is that you have a T cell that's very important, but then like with 1 week of infection or a little bit more, develop the antibody, and then you have the help of antibodies. That's possible. I know that B cell, Alan can tell you how the B cell knockouts are complicated, but I think that's the idea. So it may have a role for antibody later on. Yeah.
Later on. So using the same rationale, can you envisage that you can use the same rationale for other viral vaccines, like instead of focus on B-cell epitope.
Yeah, I think so. The problem is, I think influenza is the same. We have some results with the nucleoprotein, and it protects. And I think so because there is like, what's the efficacy of an influenza vaccine? So it varies a lot. There is some years it's 30%, some years 40%, some years it's 70%. And the problem is the hemagglutinin, it varies from variant to variant. So maybe the NP is a good choice. So someone would need to have a comparative clinical trial, but has this whole industry around the current vaccines for influenza. So there is there is another problem is all the regulatory agency uses biomarkers of protection, what has been established for the existing vaccine, and maybe they are not the best biomarkers of protection. One thing I forgot to mention is we have a model here that we infect with. We have a vaccine that induces neutralizing antibody with the ancestral, and it doesn't induce much for the Omicron. And on that model, when you use the B-cell knockout, the protection is entirely dependent on T cells. So I believe that a lot of what we're seeing, the level of protection of the current vaccines, is largely mediated by T cells.
But also you have CD4 T cell being activated.
Right.
Yeah.
Okay. Oh yeah, that's the main thing. Yeah, that's what we showed. Yeah. Okay, Doug has a question. Since I am the organizer, I'll let him.
This is a quick question.
This is the joy to be the organizer of a meeting.
What's the antigen that you picked for Plasmodium vivax, and is it a T cell epitope? Or?
Well, that's a good question. Now, for the vivax, it's basically antibody response. So it actually is strange because the vaccine induces not good interferon-producing T cells. We have some T cells that produce more like, I don't know if people still use those terms, Th2 or Th1, but it's more like a Th2 response, but induces very high levels of antibody. So we believe that the Vivax vaccine is, and the adjuvant that we use, we beginning used to use the poly-ICLC, that's an adjuvant that induces more T cells. And when we move to an adjuvant that induces more B cells, this formulation was better than the other one. So I think it's largely dependent on B cells. Any other question? Okay, thanks a lot.
Well, we will continue with the program. We will hear now from Dr. Francesco Pinotti. Dr. Francesco is a PhD in epidemiology and public health and from the Sorbonne University, and he was a postdoctoral researcher in evolutionary ecology of infectious disease at University of Oxford, and currently he's affiliated with the French National Research Institute for Agriculture, Food, and Environment. It's a pleasure to have you.
Okay, it's like 20 minutes into that? Okay. Okay. Forwards, backwards, and okay, this is the topic. Thank you. Okay, hello everyone. So this presentation will be honestly more about mathematical modeling of infectious diseases applied to influenza A, both in humans and in poultry. So in the first part, I will present some work that is not really related to vaccination, but to the dynamics of avian influenza A in Bangladesh. And then in the second part, I will present some work that I've not been directly involved with it, but I've been kindly asked to present from some of my collaborators about how mathematical modeling can be used not only to describe the transmission of pathogens, but also to generate new insights and the design of a universal vaccine for influenza A. So for the first part, let me first introduce H9N2 avian influenza virus, which is a particular subtype of avian flu that is commonly found in Asia and Southeast Asia, and also in the Middle East and Northern Africa. This subtype is normally considered a low pathogenic subtype, so it doesn't really kill chickens on its own. However, it is still responsible for substantial economic losses for farmers in developing countries, and also it can kill chickens during co-infections with other pathogens. Then there are also important public health consequences. For example, we know that H9 can lend its internal genes to other subtypes of avian influenza. This has been observed during, for example, for H7, which is a high pathogenic subtype. Some highly pathogenic strains. So in this sense, this virus can in principle allow for other strains to increase in viral fitness and gain epidemic potential. In Bangladesh, H9 is very prevalent in live bird markets, which, as the name suggests, are places where people can buy live poultry, because there are many cultural and societal reasons for why this is the case. And as I said, prevalence is very high. And in these places, in a single market, you can find chickens from everywhere. This is all Bangladesh, and the colored areas are all the districts that are supplying a single market in the south of the country. So this means that in this place you can find a large diversity of viruses, of H9 lineages from all around the country. So this is clearly problematic, because in a single place you have a high pressure, a high infection pressure with a loss of genetic material that can lead to reassortment and generate new viruses. Also, live bird markets, for this reason, lie at the interface of between humans and poultry. So these are places where we need to better understand what is going on. And this is broadly the objective of my research. So trying to understand what is happening, trying to characterize the epidemiology of this virus, and also to assess the effectiveness of potential veterinary public health interventions. So for this, I leveraged some high-quality data from the field, collected in the context of a large-scale collaboration, the One Health Poultry Hub, which was led by the UK and local teams in Bangladesh. So this study was led by a PhD student of my current advisor, Lisa Conley. What Lisa did was to put 10 chickens in a single cage in a market and leave it there for 3 days and a half, and test these chickens at irregular time intervals at 4 different time points. So 5 of these chickens come from the market and 5 of these chickens come from farms. So she repeated this experiment many times, so with many cohorts of 10 chickens and with different types of chickens that are commonly found in Bangladesh, like exotic broilers and backyard chickens. So chickens essentially that come from different types of supply chain, completely different supply chains. So my role was to try to better understand the data by fitting a mechanistic transmission model. So this model follows the tradition in epidemiology of compartmental models, where hosts are sorted into different states depending on their infection status. So we have susceptible chickens, infected but not yet infectious chickens, and recovered chickens. The model simulates also the normal functioning of this market. So we have chickens coming in every day and being sold, and also we have our test chickens that are put in a cage together with the other chickens that are sold, and these chickens can be infected because the virus is circulating in the market. there are several parameters of interest that we are interested in. One is the transmission potential of the pathogen, the basic reproduction number, R naught. Then the latent period, so the delay between infection and the onset of infectiousness. And the probability of prior exposure, which is essentially the probability that a chicken that enters the market is already infected before entering the market. So what we found was that transmission was very high in the market. So in one sense, even though I'm not showing the raw data, you could get a hint of it, because out of a few hundred broiler chickens that were tested, every chicken tested positive at some point. And whereas like 40% of backyard chickens were able to escape infection. And indeed, we estimate that if you were to leave one chicken in the market for more than 24 hours, then the probability of being infected would be higher than 80%. One main driver of such high transmission scenario is the short latent period observed in broilers, which we estimate around 5 to 6 hours, which is much shorter than the median length of stay in the market. So chickens stay in the market for about, on average, like 15, 16 hours before being sold. So this means that if a broiler chicken is infected in the market, then it is very likely that it will become infectious and spread the pathogen onwards before being sold. In contrast, backyard chickens have a much longer latent period, so we believe that they do not contribute much to transmission in this setting. So this fact alone means that the basic reproduction number is larger than 1, which means that the pathogen would be able to survive in the market, to spread indefinitely, even in absence of new infections coming into the market. But on top of this, we also have frequent viral introductions, and indeed we estimate that like roughly 10% of broiler chickens are already infected when they enter the market, and this proportion rises to 30% for backyard chickens. But there is a big difference between these two chicken types. So in the case of broilers, these are mostly recent infections occurring right before the chickens enter the market. So this means that these chickens are either latent or actively infected, so they are ready to spread the infection onward when they enter the market. In contrast, backyard chickens are mostly recovered, which means that they are immune to infection, which also explains in our context why we have this proportion of chickens that are able to escape the infection in the data. So because they were immune to start with. Then using this knowledge, we assess the effectiveness of some popular interventions, like what would happen if we were to tell vendors in the market to sell their chickens like within 1.5 days or 1 day, or what if we did anything that prevented infected chickens from entering the market. And what we find is that these two measures alone have a low effectiveness, like around 20% in the most stringent scenario. But if you are to combine them, like these are the dotted bars, they refer to the combination of these two interventions, you get a much higher proportion, a much higher effectiveness and a reduction in circulation. Then we also looked at what would happen if we were to vaccinate these chickens. So vaccination, a vaccine exists for H9N2 avian influenza, but it is not routinely used for these two chicken types, broiler and backyard chickens. However, if you assume a vaccine that is perfectly and persistently long-term immunizing, then we would get a very high effectiveness. So the take-home message. There is another part here. So what I would say here is that if we are to really want to reduce the transmission of this pathogen in this high transmission setting, we need to think about multi-pronged interventions. So a single trick won't do it. Then this is another data set collected by another PhD student, Lorcan Carnegie. What Lorcan did was to go into markets this time and sample a few birds from 2 stalls in each market every other day for 1 month. He then tested these chickens for the presence of H9N2 influenza, and he continued to sequence the positive samples and produce a phylogenetic tree. So here the colors refer to the markets where the sequences were taken. So this is like a very nice information, because using such a tree, we may be able to better understand the epidemiology of the virus. In fact, we know that it is a well-known fact in the field of phylodynamics that the dynamics of a pathogen affects the shape of the tree. So by reverse engineering this fact, we may be able to infer something about the dynamics of the pathogen starting from the tree. So this has been the question that I've been grappling with for the last few months. So we want to learn something from this tree. We want to learn why the sequences seem to cluster by market. What are the processes behind? What does it mean about the turnover of avian influenza lineages in the market? And what are the drivers of lineage persistence? So like how long these lineages persist in markets. So to do this, we used a coalescent-based approach that synthesizes information about viral movements between different viral populations. Here we have 3 populations, M1 and M2 refer to the 2 markets in our study, whereas A is a so-called ancestral population. You can think of it as farms. These are this is the population, the idealized population, where the viruses where viruses are born and then are moved to the market because the chickens are moved to the market to be sold. So the outcome of the coalescent process, I'll be very brief, is a phylogenetic tree. So every internal node in the tree is called a coalescent event and represent an ancestor, a potential unobserved ancestor in the past, and therefore a past transmission event. We don't know where this transmission event occurred, but using a model we can try to infer that. So, and try to see if transmission happens in one of the 2 markets or in the ancestral dim. And what we found was that essentially there are very few coalescent events that occur within markets, like less than 5 likely. And for reference, the maximum value would be up to the number of sequences, which is about 60 for this dataset. So this means that the diversity within the markets of avian influenza virus is dictated by dynamical processes that occur outside markets within the ancestral population. So, and this is essentially because we have a very, as I showed in the first part, we have a high frequency of viral introductions. So essentially, it is very difficult that if you sample a few sequences in the markets, that you are getting one descendant of the other of another sequence. Then we also implemented an individual-based model to characterize the fate of each individual introduction, which we call a lineage. So every infected chickens entering the market is a new lineage. So we, using realistic parameters that we infer from our previous analysis, we found that every lineage persists for about 1 to 3 days. And so this is a very short time. So all these transmission chains are very short. And the reason is that we have lots of introductions, so lots of competition for susceptible hosts. So many lineages will go extinct eventually. We related this persistence time to different properties of chicken trading. For example, the color here represents the probability of a chicken remaining unsold after 24 hours at the end of the day. So the darker the color, the longer the chickens stay in the market. As you can see, a longer stay in the market promotes the persistence of viruses. So the conclusion for this first part is that high level, we find high levels of H9N2 avian influenza virus transmission in live bird markets. Different chicken types have different epidemiological properties, for example, in terms of latent period, but also the history of exposure is very different and depends on the way chickens are handled before reaching the market. Then I believe that we need multi-pronged interventions if you really intend to reduce the burden of this virus in the market. And I believe that genetic data is very helpful to understand the dynamics of this virus. Okay, so now on to the second part, which is more in line with the spirit, the theme of this session on vaccination. So this is work that has been performed by some of my collaborators at the University of Oxford, Sunetra Gupta, Jose Lorenzo, Hani Zinada, and Craig Thompson. So this is long. I'll do my best to present this work, but I will accept any questions later. So this theme, this line of research has a long history dating back from some work, theoretical modeling works by Sunetra Gupta from 1998 onwards. That now led to the design of some universal vaccine for influenza A in humans, not in chickens anymore. So the rationale is that current vaccines against seasonal influenza target epitopes that are naturally protective and are found on the head of the hemagglutinin protein, which is one of the surface proteins of influenza. But the issue is that these epitopes are also highly variable due to the accumulation of point mutations. So this means that the vaccines that are based on these epitopes are very effective against this specific conformation of the epitope, but if a new virus with a slightly different conformation appears, then the vaccine will be less effective than that. So such a vaccine cannot be called a universal vaccine. On the other hand, it doesn't make sense to target epitopes that are not variable at all, but that are not immunogenic. So what if we could find epitopes that are both protective and of low variability, so to prevent the possibility of new variants appearing anytime soon, and reducing our effort, the effectiveness of our efforts. So in 2007, my co-authors published a theoretical paper, where they defined a model, an alternative model of influenza transmission and evolution that supports the existence of these epitopes. So this model is a sort of an alternative to the prevailing antigenic drift model and assumes that the transmission dynamics of influenza A is described by a competition between a finite number of antigenic types. So an antigenic type is a combination of antigen epitopes that may be more or less variable. So here I'm making a very simple example of one antigenic type that is characterized by three epitopes, sorry. So the circle one is the most variable. It has 5 different conformations, 5 different colors. The square one is moderately variable. It has 3 different variants. And then we have another epitope that is not so variable. It has only 2 variants. So in this case, we have 5 times 3, 15, 30 potential antigenic types that are competing with each other. And so the transmission, the dynamics of this virus, is characterized by the sequential emergence of different types depending on the landscape of immunity against each different epitope. So the advantage of this model is that it can explain the chaotic dynamics of influenza A, but also some other features of, for example, the fact that how antigenic distances evolve in time in sequential epidemics. So in the end, in 2017, they found one such epitope of low variability that is also highly immunogenic. Interestingly, this epitope was called Oreo and is related to H1, influenza A. So they found then by analyzing historical isolates of influenza A strains, they found that from 1918 up to present, up to present, we have observed like 5 different variants at this of this epitope that are named like blue, red, orange, green, and pink. And they were able to do some tests on a model with based on mice. So they tested like a vaccine. They did a bunch of neutralization tests, and they found that mice that were vaccinated with a red Oreo from 2006 or a green variant from 1977 were able to neutralize themselves, each other. Red and green are very similar to each other, but also strains from 1933 and 1934. But they were not able to neutralize blue from 2000, that reappeared from 2009 onwards. In contrast to the blue the blue mice vaccinated with blue were able to neutralize the Spanish flu-like strains from 1918. Finally, and then we'll stop. We were also able to do like some challenge experiment on the mice, and they found that mice that were vaccinated with red or green were and then challenged with a strain from 1934, which was like red-like, were quite protected against it. So suggesting that these neutralization results translated into cross-protection against a strain that these mice never exist. And similar results were also found in the context of children's sera from 2006. So to conclude, so we have a mathematical model that suggested the existence of epitopes of limited variability. Immunity against these epitopes shapes the antigenic evolution of influenza A, like the sequential appearance of distinct antigenic types, because as we've seen, like red was present in 1934, then disappeared, and then came back later in the 90s and the 2000s. Or blue was present in 1918, and then was present again from 2009. These epitopes of limited variability are potential candidates for universal flu A vaccine. Okay, thank you for listening. One more?
Because it was the last. Yeah, but it passed. I think they removed. Okay. You have one last slide. Okay. Just a question. Do we have any questions for Dr. Pinotti? Yep.
Maybe I missed the information, but about the epitopes.
Yes.
You insert them on the HA, or you just use the epitope to induce the neutralizing antibody? You use a peptide, small peptide. What's the? I was just confused about.
Yeah, I think
it shouldn't be a conformational epitope that induces the neutralizing antibodies again.
Okay, so unfortunately, I'm not an immunologist, so I'm not sure if I can answer this question. So is your question more about like how the vaccine was then implemented in practice? So so from my understanding, it is like three-step vaccine. So I think it was like the first 2 rounds were made with a DNA vaccine, and then there was probably a peptide inserted on an lentivirus.
But it's just a single peptide, or it is the HA exchange?
So they put the HA. I think they put the HA on the virus with a conformation, but the HA was not the H1. I think it was H5, H6, and probably, I don't know, maybe H11, but I'm not sure about that. So in order to not create any, I think the rationale was not to create any specific response to any other H1 epitopes, but just to, in particular, to this epitope of low variability. So I didn't mention that, so it didn't.
Thank you to Dr. Pinotti. Continuing, we will listen from Dr. Santuza Teixeira. Professor Teixeira is a biologist and holds a master's degree in molecular biology from University of Brasilia and a PhD in biochemistry from the University of Lausanne. He's a member from the Brazilian Academy of Sciences and is the vice coordinator of the Center for Technologies in Vaccines and a full professor at the Department of Biology, Biochemistry and Immunology of our university. It's a pleasure to have you.
So, good afternoon, everybody. I'm very honored to be here to talk about some of the work we are doing at the Centro de Tecnologia de Vacinas, CTVacinas. I thank Ricardo and Luis Alcântara for inviting me to show this project. And actually, I must say, these are projects that are in the early stage of development. So I'm going to show most preliminary data, non-published data. And I think it's important to say that most of the data I'm going to show you are being generated by graduate students working at the CTVacinas. And of course, I don't need to tell you how important RNA vaccines became in the past years. I'm just going to remind you a little bit of the history of the mRNA vaccines. We need to go back to all the studies that have been done in the early last century, since 1960, on RNA structure, RNA function, and that's all was very important for getting to the RNA vaccine during the COVID pandemic. So can go back to start with Jacques and Monod, that imagined there was such a molecule that later we found out they were the RNA. And also Kathleen Caricot and Drew Weissman that showed that it's very important to use modified RNA if you want to develop a vaccine. And the capacity of preparing nanoparticles, lipid nanoparticles that can solve the problem of delivery RNA vaccines, of course. These are some important milestones. And this is just a nice graph. I always like to look at that because it shows what happens in Israel during the pandemic when the vaccine started in December 2020 with the Pfizer vaccine. And very quickly, so all the number of cases start to drop even after the government stopped to do shutdown. So it's, of course, it's a very, very efficient. And then based on all of these, we decided, like about 2 years ago, can we start setting up a platform for RNA vaccine at CTVacinas? So it's very nice that Ricardo already talked about how we structure the CTVacinas. So we have different platforms, different groups working on different types of vaccines. But independently of the vaccine you're working on, you are working mainly on the step of making RNA or on the step of producing a recombinant protein. So this is work mainly as a fabric and not a real, academic lab. So we decide to implement a platform for RNA vaccines and see if it works. And these are showing that we can start from the proof of concept, validate preclinical studies, and then we hope we can get to clinical tests with these RNA vaccines. And I'm going to talk about the project on RNA vaccine for leishmaniasis and for Chagas disease. And in the last few slides, I'm going to show quickly some data on RNA vaccine for dengue and influenza also. Since this is the first time we are talking about the parasitic disease, I'm just going to make a very quick introduction on leishmaniasis. So it's still a major health problem in countries like in Brazil. About 12 million people infected everywhere in the world. Brazil accounts together with 5 other countries, for 90% of all the cases of visceral leishmaniasis. There are 2 main types, visceral leishmaniasis and cutaneous leishmaniasis, and at least 20 different species of leishmania that cause the disease. And it has a quite simple life cycle depending on the vector, which is the phlebotomine or the sandfly, and the intracellular cycle of the parasite in the mammalian host. And Chagas disease, that's also still a major health problem in Brazil. Much less cases, around 6 to 7 million people still infected. Mainly 2 clinical forms when the disease evolved to a symptomatic form. It's mainly intestinal or heart symptoms. Even though we had good success on vector control, which is depending on the triatomine bug, we still have learned more and more new cases of Chagas disease due to oral transmission and also congenital transmission. And treatment of Chagas disease is still a major challenge. So, of course, I'm not going to have to explain how RNA vaccine works, but it's important to show that because the RNA vaccines induce a very potent cellular response. This is very important, not just for leishmaniasis, but also for Chagas disease. If you want to have good protection, you have to have a good cellular response. And then you have to go to these 3 main steps: mRNA synthesis, encapsulation in LNPs, and of course, different immunization protocols. So if you want to make RNA, you need to produce a molecule that is functional. So of course, you have to produce an RNA that contains the coding sequence for your antigen, 5' 3' UTR, poly-A, tail, and a 5' cap modification of the RNA. And one of the main improvements was to use modified pseudouridine. That's what we use in CTVacinas instead of uridine. And we use a reaction that makes the addition of the cap modification during the in vitro transcription. So if you decide to start making RNA for one particular vaccine, one particular antigen, there are several questions you need to think about. Which kind of 5' and 3' UTR sequence you're going to use, because this will impact both the stability of the RNA and the translation. Coding sequence, are you going to modify the codons? Because if I'm trying to express a trypanosome or bacteria antigen in a mammalian cell, do I need to make codon optimization? Do we need to include the signal peptide if you want to make the protein to be secreted by the cells or not, or to be produced in the cytoplasm? And another question is, do you need to purify the double-strand RNA? Because we use in vitro transcription reaction with RNA polymerase that end up producing a significant amount of double-strand RNA that may inhibit the translation of the RNA. So once you decide what you're going to do, you prepare your template with your coding sequence and the T7 promoter, and then you generate your RNA, but then you need to encapsulate in the lipid nanoparticle, and that's another step that we work a little bit to decide to make some tests using different types of lipids. So you have 4 main types of lipids: cholesterol, a pegylated lipid, and a phospholipid. But what is really important is the ionizable cationic lipid that interacts directly with the RNA. And of course, this is an issue of intellectual property, because if you use the Moderna or the Pfizer ionizable lipids, you end up with protection by patent. So what we decide to do is to try to make a LNP using some other type of ionizable lipid that we don't need to run into this question of PI. So the way we go to test our different RNAs is after you do in vitro transcription, we have to verify everything works okay, and then we encapsulate, and then we look at the quality of the LNPs, if they have the right size, and if they actually are protecting the RNA inside. So after you encapsulate, we run again a gel to see if the RNA is still in the right size. And then, using luciferase RNA, we start to looking for different LNPs, changing the lipid composition. And then here we just show one experiment where you're comparing a luciferase RNA delivered with a LNP with the SM102 lipid that is from Moderna vaccine, compared to another lipid that we select in CTVacinas, and we see that they both work very well in the animal. And also we show this is delivered mainly to liver and spleen. And we also show that using the lipid either with the RNA or without the RNA, it's also a component that induces an immune response because the LNP will work as adjuvant in the RNA vaccines. So this is shown by measuring inflammatory cytokines in macrophages that were incubated with these LNPs. So then we decide, well, that's the main question, which antigen we are going to use for this type of pathogen. The first antigen we test is a protein that we heard about yesterday. It's a protein identified by Ana Paula's group on Leishmania infantum protein that it's been used to develop the Leishmania vaccine that Ricardo just mentioned to you. And it's called LINCAP protein. It's a protein associated with the mitochondria of the Leishmania, and it's highly immunogenic, as shown here. But in addition of the fact that this protein is expressed both in amastigotes and promastigotes of Leishmania, we select this protein because it's highly conserved if we compare with sequence from other Leishmania species. So we may be able to use this sequence to protect against different species of Leishmania. So the way we go then to test to see if this protein, this RNA sequence will work, we do prepare the LNP with the RNA, and then we are comparing with the recombinant protein, which is the work published by Bianca in CTVacinas, showing that the recombinant LINCAP protein is also efficient in protecting mice and hamsters. So what we decided to do is to make a LNP with the LINCAP protein and then compare the results of the protection of the RNA compared with the protection when you use the recombinant protein. So this is what we have to do before we inject the animals to make the LINCAP protein, look if the RNA is okay, and then we transfect cells with the RNA to make sure that it's functional. It can generate the protein inside the transfected cells, and also we look by immunofluorescence. And then we did a protocol with 3 doses of the RNA, 3 doses of the protein, and then we are getting about similar immune response compared with the RNA and the protein immunization. Even though with the protein we get more IgG1, and then with the RNA we're getting a little bit more IgG2, which is a good sign. And then we measure interferon gamma. So what we are seeing here is like you have some good production of interferon gamma with either with the RNA or with the protein. And then when we do this is done in bulb C mice, and then when we do the challenge with Leishmania infantum, so we are having the count of parasite burden using limiting dilution of spleen and parasites present in the liver. And then we are comparing actually here if we do the immunization using the LNP containing the sequence. either with the Moderna lipid or the lipid from CTVacinas. So we are getting similar results too. So we are doing a number of experiments trying to combine different protocols of immunization, and one of these tests we are doing is that what if we combine protein immunization with RNA immunization in a heterologous protocol, and that's shown in this slide. Here we use 2 doses of mRNA and 1 dose of the protein, or 1 dose of mRNA and 2 doses of protein, or only proteins. And actually the results shows there is no big difference. There is similar partial protection if we use these 3 different protocols. We have good IgG and IgG1 and IgG2 production. But we have very small production of interferon gamma compared if we use only the RNA compared if we use RNA mixed together with the protein, which was kind of surprising here. So that tells us that we still need to make some more tests with different protocols, but also using different antigens. And that's what we are doing right now. Including some other different antigens for the leishmania vaccines. So if I move to the Chagas disease vaccine, Ricardo also mentioned that in CTVacinas we are pretty much advanced in the project to develop a therapeutic Chagas vaccine using a chimeric protein that shows good protection if you immunize mice with this chimeric protein that is a fusion between 2 different isoforms of the transialidase of Trypanosoma cruzi. Transialidase is a surface protein for T. cruzi, which is very abundant in the surface of the bloodstream stage, tripomastigotes. And it's responsible for transfer sialic acid from host proteins to the parasite surface. And this is being used as a target for different preclinical and proof of concept experiments on Chagas disease vaccines. So we decided also to concentrate on the transialidase to start. And this is important to show that these transialidase that have this catalytic activity that are exposed at the surface and do the transfer of sialic acid, they, in addition of the catalytic domain, all these active transialidase contain a C-terminal domain that has several B-cell epitopes because it's encoded a number of repeat amino acid of about 12 amino acid repeats. In different copies of the transialidase, you have different numbers of the repeats in the C-terminal domain. And previous work in my lab, we have shown that if we produce the transialidase with the full-length sequence, the sequence that has no C-terminal repeat domain and only the repeat domain, and if we immunize mice with the full-length protein, we're getting antibody response against, of course, the full-length protein. But then we get lots of antibody against the non-catalytic domain, which is the repeat domain of the protein. So the question we ask here, if you're going to make an RNA vaccine use the transialidase protein, should we include or not the repeat domain of the transialidase? And we got even more curious about this because if you immunize mice with the recombinant protein, we see a major difference when we immunize mice containing the protein without this repeat, so we get a better interferon gamma response. And however, the protein that contains the repeat, the full-length protein, we have a better IL-10 response production. So we decided to prepare RNAs with the transialidase sequence, including the catalytic domain only or the protein containing the full-length protein. So we produced the 2 types of the RNAs. encapsulated the two types of the RNAs and did the transfection in I think this is HeLa cells. Then, as I showed before for the Leishmania antigen, we have to show that the RNA is actually producing the protein in the cell. So this is the full-length protein. This is the protein without the repeat domain. And then we immunized animals and showed that you get a very good response if you use the full-length protein. And then we get much less antibodies if we use the protein without the repeat domain. And now if we look in the splenocytes for the immunized animals, they were re-stimulated with the full length or with the protein without the repeat domain. Again, we are seeing kind of the same level, the interferon gamma production for the splenocyte. But the animals that were immunized with the full-length protein, we get a better IL-10 response. And why is this important? If we now go to this challenge with the Y strain, that is the virulent T-Cruz strain, and we use a Y strain that is expressing luciferase, we don't see very much difference in the protection levels if we use the full-length protein or if we use the protein without the repeat domain. And both using the counting parasite in the blood, using the bioluminescence signal, also counting DNA in the heart tissue. Here we do see a difference in the DNA in the tissue. But what was interesting was when you look at the heart of these animals, the animals that were immunized with the full-length protein, we are getting much lower inflammatory response. So which is, in terms of Chagas disease, that will be an interesting outcome to have lower inflammatory response due to the vaccine. So this is the results I'm going to show you about the Leishmania and the Chagas disease vaccine. And now I'm just going to show you very quickly, I think 2 slides about a dengue vaccine. And this is the work done mainly by Sarah. She's a student doing her PhD. And what she did first, we decided to test the RNA based on the non-structural protein NS1 of dengue. We also have this test using the envelope protein, but I'm showing the result with NS1. So she produced RNA encoding NS1 for 4 subtypes of the dengue virus, and she did just the same, so transfect HEC cells and show that they're producing the proteins using the RNA that she in vitro transcribed. And then she showed a good antibody response using each one of these proteins. And then she showed that if she mixed these 4 RNAs in one formulation and immunized animals, she gets a very good protection if she challenged with the dengue 2 virus. So that's one of the big advantage of the RNA vaccine. You can mix together several sequences instead of making 4 different vaccines. And then you have a very good survival of these animals that were challenged with dengue virus. And finally, this is the results using influenza H5N1 nucleoprotein RNA vaccine. The same protocol was followed, so we do the in vitro transcription, transfecting cells, see if the cells are producing the right protein in the transfected cells, and then immunize animals. These are 2-dose protocols. The animals show good IgG levels, interferon gamma production, and then when she did the challenge, she also observed a very good protection, both when we count parasites and when we count, when you measure the weight of the animals. Also, histopathological results also show the protection of the lungs of the animals that were immunized with the LNPs containing the RNAs for the nucleoprotein. So I think this is the summary. I guess I'm pretty much on time. We can show that we are using different protocols for using Leishmania antigen. For the T-Cruzi antigen, we defined that we're going to work with the transialidase containing the repeats. And for the dengue and the influenza vaccines, we are kind of a little bit more advanced, going to the scale up, trying to how we're going to scale up production to make this clinical test with these vaccines. So I need to thank people that work on this project. The project, Gabriela and Renata Salgado, they are mainly responsible for producing RNA sequence in vitro transcribed for all types of vaccines for every project. Renata, she's very help with the production of the LNP formulations, changing the lipids, and trying to develop different LNP protocols. These are the people that work on the parasite vaccines, Leishmania and the Chagas, Ana Flávia, Nayuma, Carlos, and these are the ladies that work on the viral RNA vaccine, Sara, Natalia, and Lydia. And this is all done under really the guidance of Ricardo, Fabiana Machado from UFMG, Ana Paula, and Diego from UFMG, and Edson Dorigon from USP. I think there's probably many other people involved that I'm not going to mention here, but we work as a team in CTVacinas, so all these studies have been done with a lot of other people involved. Thank you for your attention.
Do we have one question to Professor?
That was great. I have a quick question for you. So when you use the sialidase as an antigen, it looked to me like when you use the full-length protein, it looked to me like you had no parasites in the heart muscle, but you still had inflammation. I didn't quite understand why that is. Is the inflammation due to the vaccine, or is it due to the parasite?
I think it depends on the timing that we did the qPCR and the time that we did the histopathology. I'm not sure. We still get a little bit of parasite in the heart. So I think that's just a matter of the amount of DNA we're detecting, the amount of parasites.
Okay, thank you, Professor. We will have a very short coffee break. We'll have a 15 minutes, and we'll be back. Thank you.